हाँ रिकॉर्डिंग होता है।
Assalamu alaikum everybody, and good morning and good afternoon, whichever is applicable according to different time zones, and welcome to the fifth session of this very informative training course. Our current session will focus on current trends in halal science research and innovation, and our next speaker is the chief coordinator of this training session, Professor Dr. Sayyid Ghulam Musharraf. Professor Musharraf is a well-known scientist of Pakistan in the field of mass spectrometry. He obtained postdoctoral in Australia and USA. He has published over 180 research articles with a cumulative impact factor of over 550 and one US patent. Due to his scientific contribution, he has been awarded with several international and national awards and honors, such as recipient of Fellow of Chemistry, Society of Pakistan, selected as a member of Global Young Academy from Pakistan, selected as the third World Center Academy Sciences Award. He is also serving as the editor of many international journals, including the scientific reports. He has supervised over 60 PhD and MPhil students in the field of mass spectrometry. So over to Professor Musharraf now.
Assalamu alaikum. Thank you, Dr. Shakil, for your kind words. May I have a presentation, please? Okay, so in my talk, I would try to cover some aspect of halal research that my research group is doing currently. But in addition to that, the two important things that I'm trying to cover in this talk, the one part, a very small part, would be related to kind of motivational words, because that how I entered in that field is important to know, at least to share at this forum. Because initially it was not planned to involve in halal research and all these things. So that's important to know, especially for the young scientists and for the upcoming students or the students who are working in food science related. Obviously, the second part would be just to introduce that because since yesterday you are listening about particular name, that is mass spectrometry, which has indeed a strong application not only on food science but in the biological sciences as well. So I'm going to talk about a little bit some important application of mass spectrometry. And the third part, as I have mentioned, that is about the analysis of food samples that are currently being done in my research group, followed by the some important work that we are doing in halal research. This is my field of interest. You can see a lot because yesterday someone asked me that usually in Pakistan we don't focus on a particular issue. We are too diluted. I totally agree. Especially scientists from Pakistan, most of us are diverse. I mean, we have to. Maybe that our upcoming generation would be more focused, and the reason is very simple. Unfortunately, we don't have a kind of scientists required in each field. The field is expanding tremendously, but in contrast, the capacity that we have in Pakistan is limited. So every individual has a kind of 4 or 5 assignment to cover, to address. So that is the reason that Pakistani scientists are diverse and they are doing so many things or try to accommodate the things as much as possible just to address that we are there. We should cope up with the current scenario of world trade. Like we all know that the artificial intelligence nowadays is quite in research. And in Pakistan, the people are doing, but it's a force. Everyone is asking that where is the AI component in your research? Do you have an AI component? So these are the things that we are currently facing. So by discipline, I'm an organic chemist. I did my PhD related to kind of a structural elucidation. During my postdoc, the institution has asked me to develop the facilities and the expertise in the field of mass spectrometry. Believe me, at that time, I just know after my PhD that what mass spectrometry is, and it's a background, that's all. So since then, I'm trying to learn what mass spectrometry is and what's its application. So they asked me to learn about the mass spec. So during my postdoctoral study, that what I've tried to focus on mass spectrometry. And now, Alhamdulillah, I'm the first one. In fact, I'm pleased to share that because I'm the first one in Pakistan who established the metabolomics mass spectrometric research in Pakistan, So that is the thing, that how you entered in the field and cover the needs of your country. So after learning the mass spec applications and the different instrumentations, we have applied this research in various fields. The first one, as you can see, that the phytochemical analysis or pharmaceutical analysis, because Pakistan, is most of the people are using the herbal products, for different purposes. And we have a very strong herbal companies like Hamdard, Karshi, Herbion, and we all are using their products. But these products are not getting into the foreign market effectively because of several reasons. But one of the reasons is that the standardization is missing, the value addition is missing, the data, the chemical and biological data is missing. So this is the slot where we are contributing, that how the mass spec can contribute to handle the standardization of herbal products and the value addition. So we are working on with the Herbion and the Hamdard and try to standardize their products with the help of mass spectrometry and make some value addition. So this is the part where we are doing. The second part, actually, I went to US in 2007 for my another postdoctoral study, and at that time, because being a chemist, I have no idea that how you can handle the disease-based biological samples. So that's what I have learned from US, especially the proteomics analysis, the biomarker research. So, since then, I'm working on the different application of biological mass spectrometry, particularly biomarker discovery and validation. And my research group is extensively working on the beta thalassemia treatment, the students who are facing blood disorder like beta thalassemia, so how we can cover up. So this is a kind of treatment most probably that we are going to have in next 2 years. And inshallah that it would be launched in Pakistan, some new drugs for the treatment of beta thalassemia. So this is the one of the biological part of my research group. The third part, actually, no one has asked me to work on that side. Why I've entered into the food agriculture side and all these things. Because I remember that I visited Faisalabad once around 6 to 7 years before. So then I realized that we have a very good agriculture scientist. But the issue which they highlighted me that they are lacking an understanding, the molecular understanding of the compounds, which needs to be identified first. So where we can play our role. I mean, obviously that with the help of mass spectrometry, you can identify a phenotype, you can identify a molecule which can be linked with the biosynthetic pathway, and that can be linked with the genome and the whole pattern. So then I've realized that we need to enter into the food analysis, and since then, we are working on the food and publishing so many papers. So that was related to food part. Now come to the halal stuff, because being a mass spec scientist, I remember that a man visited us around 6 or 7 years before. He was from Agilent. He was a kind of scientist working on the different application of Agilent mass spectrometry. So during our discussion, the discussion turned over towards the research, and fortunately he was Muslim. So then he actually explained me that the needs of a Muslim scientist in halal research, and he highlighted me 2 or 3 important issues. And he asked me, Musharraf, why you are not entering in that field, and please address this issue as well. I'm already working on the food analysis. Then I've started to work on and realized that indeed there is a need of a Muslim scientist to work on the issues that are relevant to our religion, Islam, because we should not expect that someone else going to work and address that, okay, that this pork is really not good for health and should please stop it. This alcohol is not good for your health and please stop it. It's always a controversial issues. So we need to address these issues by ourselves and explain the things in a language of science. So actually that discussion motivated me, and then I've entered in this field. Since then, we have started to work on the halal science, and alhamdulillah, in last 3 years, somehow I represent my country as a scientist at the international forum. I'm the one who have presented the work at the CIMIC forum, Halal Assembly, Thailand, Philippines. So the different assembly, in the different halal assembly, I've presented this kind of work. And because of our contribution in halal, the CIMIC has appointed me as one of the technical member from Pakistan. So the reason to explain all this is that the things just to inform you that you can also do the same thing. Okay, because if you have a molecular background or pharmacy or whatever, you can switch, you can take a challenge, and it is doable. आज से 10 साल पहले अगर मैं ये सोचता तो शायद आज मैं यहाँ नहीं होता, और ये मुकाम पे अभी जो बातें हो रही हैं ये शायद ये भी नहीं होता. So initially I was thinking that why we can't. So since I've started, and now alhamdulillah we are doing such type of things. So that's a brief story about as a kind of motivation that you can enter in that field. Nothing is impossible, believe me. So you can work on that. The title of this session is the current trends of halal research, and you can see that the omics, the word omics, I'm sure that those who have science background, so they are much familiar that since last 2 decades, the people are extensively work on the various omic sciences, genomics, transcriptomics, lipidomics, metabolomics, metalomics, you can just name it. But why they have started to work on this omic sciences? The reason is very simple, because the innovation of instrumental facility. There are instruments available by which you can quickly genome the whole human genome. The Professor Ataur Rahman was the first Pakistani whose genome is publicly available, and that work just done in few days. Because of the advanced instrumentation since last few decades, the huge transformation of the instrumentation is already in the market. Availability of databases, because every time you need to identify the DNA or protein or metabolites, and obviously you need to have a databases. So drastically improvement in databases. So that's why the researchers have entered to the omics side. A statistics tool, because you always need to compare that upfold change or downfold change or quantitative threshold or qualitative threshold. So the statistical tools are in the market. So these are the reason that why people since last few decades have entered into the omics sciences. So if you're going to talk about the metabolomic side, the metabolomic is something which is very fascinating and easy to handle. The metabolomic is the phenotype of your gene, right? Whatever the gene have command and instruction, ultimately it would appear as a production of a small molecule which term as a metabolites, right? For example, and they are very useful. We all know that, because for example, in case of diabetes, what we all need to monitor? Just glucose. The sugar is a small molecule, a clear indication of your disease status. It's a phenotype. No one going to screen a gene. So the gene is quite complicated to screen. So the phenotype, a small molecule, somehow easy to detect because they are high in concentration. And the similar, in the plant sciences, in the animal sciences, in both, I mean, the things are common. Addition to that, the metabolomics somehow present a limited number of molecules that can be covered. Especially if you are talking about the human metabolome, the numbers are limited in comparison with the proteome or genome or transcriptome. They are huge in number. So metabolites coverage comparatively easy, and you can easily handle that. The techniques available to handle the metabolites, yes, NMR, mass spectrometry, and some other techniques as well. But we will talk about more in detail about mass spec, that why we need to have a mass spec for such type of analysis. Apart from all this technique and advancement, the metabolomics sample can also be handled easily. As far as the protein samples concerned, it's really difficult to handle if you are going to analyze a protein sample, the stability and everything. The same thing is also in metabolite, but somehow, for example, in case of plant metabolites, the stability is not that important. They usually are a stable compound, and you can easily process them and extract and analyze. So this is one of the advantages if you are going to talk about the metabolomic side. So this is the comparative comparison of a different omic sciences. So the metabolomics, as I have mentioned, is actually giving the current status of a body, of an animal, of a plant. It could be anything, animal, any type of source. So it gives you the current status of your body. For example, if you are going to test your sugar, and if it is normal, which means that you are non-diabetic, it's very clear. You have a normal, healthy body status. Okay, so this is the kind of current shot that you can quickly monitor. A set of, it could be a targeted metabolites, it could be a set of metabolites, because in the next few slides you will see that when we are going to talk about the research samples where we have compared a set of metabolites. Okay, so it could be a set of metabolites or a single metabolites comparison, like as I've mentioned in case of diabetic. So now is this few slides, especially next 2-3 slides, for the students or researcher who are using or planning to use the mass spectrometry. Why mass spectrometry is important? It's a simple question. The reasons, so many. So one of the reason, the mass spectrometry have a good combination of form available as a hyphenated technique. What does it mean, hyphenated technique? Hyphenated technique means that you can couple two techniques together, like HPLC and mass, just combine them. It's an HPLC MS. The first part gives you the separation, and the second part offer you a analysis, right? This is an hyphenated technique. GCMS, the GC covers the analysis of or separation of compounds, volatile compound. Mass covers the analysis of the compound. So this is the hyphenation techniques is available. And this is indeed and the only technique which gives you a true hyphenation approach. Because the people are talking about the NMR, LC NMR, you know, but unfortunately it's a failed technique. As we're talking about the LCNMR, you cannot run the LC with deuterated solvent. It's not a true and never be a true hyphenated technique. It has to have some pause in between, right? Similarly, some other techniques, but if you're going to talk about the mass, so then you can link the GC-MS, HPLC-MS, capillary electrophoresis MS, LC, ICP-MS, you know, TLC, MS. So there are so many techniques commercially in a true forms are available where you're going to talk about the hyphenated one. Hybrid, what does it mean? The hybrid concept is also available in the mass spec. We all are familiar with word hybrid. कहाँ पे सुना है hybrid? Hybrid क्या आगे? गाड़ी देखें सबसे पहले गाड़ी. So we all know the hybrid cars are available. So hybrid car, I don't want to discuss the hybrid car, but you know the technology which gives you a better fuel consumption and safe and secure for your environment as well. So similarly, in case of mass spectrometry, you can couple more than one mass analyzer. You can couple one quarter pole, second quarter pole, and triple quarter pole. The triple quarter pole mass analyzers available. You can combine the quarter pole with TOF, time of flight. Okay, so you can just name it. So all types of combinations available in the market, and the beauty of that hybrid form that you can identify a compound with very high resolution. That is the advantage of the hybrid form, right? You can identify the compounds with very high accuracy and sensitivity. That is the advantage of hybrid form, right? So these are the features. So that's why the people are using a different hybrid version of mass instruments, and it's commonly available. The last speaker, if you just look, she mentioned about the quarter pole ion trap, you know. So that was the two mass analyzer combined, and with the help of that feature, they can perform the MS analysis of an individual, of a small molecule, of a protein. So that is the hybrid form. Yes, the third feature is ionization technique. Is a complete buffet. We personally, I always like to go a buffet dinner because you can eat whatever you want. So similarly, the mass spectrometry offers you to have a complete buffet of various ionization technique, and you can select it based on the polarity of the compounds. A compounds or the molecule which you want to analyze is you can select accordingly. For example, in case of protein, we all know that's quite polar. The best option would be electro spray ionization. Okay, the technique is there. If you are going to talk about the aroma analysis, a volatile fragrance analysis, the EI, the electron impact, that is available. If you want to talk about the medium polar compounds like natural products, the APCI is there. So all types of molecule can be analyzed with the help of mass spectrometry, and that's why it's a universal detector. Okay, so this is one of the important features if you are going to work on the mass spectrometry. You know why it's important to know? Because then you can defend and learn that why you need a mass spectrometry in your center or in your testing laboratory. It's really needed. Believe me. अगर आप कुछ इस पे PC1 वगैरह लिखते हैं, just include that mass spec in your budget. It's really needed for all types of application related to biological sciences or non-biological sciences. So these are the things, the most important thing. The other advancements are available. That, for example, the techniques available like RIMS, DART, and ion mobility. You know, especially in US, what doctors are doing nowadays, they are using a mass pack during the operation. Can you believe that? Because particularly the breast cancer operation, they need to know exactly the location of extended tumor region, and they have a kind of pen, simple pen, which has a probe, and they use to burn the cells and identify the cancerous marker. And with the help of this is during the operation, and during the operation they exactly identify the region of cancerous part, and then they remove it. So now you can see, this is the RIM technology. So now you can see that the application of advanced mass spectrometry in the real analysis. Similarly, जो biological आपकी testing होती है, blood culture, you know, if you are going to talk about the blood culture analysis, आप कहते हैं culture करा ले, so it takes how many days in a trivial analysis? 2 to 3 days. If you want to culture your blood sample, you have to wait until, you know, till they get the result. You have to manage your pain, because they would not assign you the right dose for the treatment, because they need to know that what exact infection you have. You know, the MALDI biotyper is in the market nowadays, and the modern world are using those instruments, and within 30 minutes you can get your data and can be treated. So this is the kind of advancement the world is actually enjoying by the mass spectrometry. But the most important feature, which I always highlight in all my presentation, is nothing but its sensitivity. The mass instrument is a highly sensitive technique, and you can detect the molecule up to, if you just look, this slide is hard to believe. It's 480 molecules, and which is equivalent to 800 yeptomole. Not femto, not, you know, it's an yeptomole molecule sensitivity, which is unbelievable. However, the routine analysis of mass spectrometry can be range of Ng to Pg. You know, this is the femtomole analysis. So that is the beauty of mass spec, and that's why mostly research related to biomarker or the metabolites and everything is based on the mass spectrometry. Dope testing jo hota hai, you know, the dope testing is a kind of testing which can only be detect the dope compound with the help of mass spec, because usually they used to dope, the sportsman used to dope a very low level of steroids or anabolic compounds. So the mass is the only one which can detect it. So this is about the features, and just a comparison of LCMS and GCMS. Why I've included, because most of the research related to halal is the people are using either LCMS or GCMS. So you can look in that picture, the GCMS somehow is indeed the good one, but it has a limitation of compounds nature, you know, which actually related to the volatility of the compounds. And the compound is less polar or somehow medium polar, so you can separate out on GC and then analyze with mass spec. In comparison, LCMS, you can see it has a broad coverage. You can analyze a very small molecule like sugar, but must be polar enough and must be, you know, soluble in a polar solvent, and can analyze a very large molecule like protein, peptides, and DNA. So the LCMS nowadays are getting more attention because it covers wide range of analysis. But GCMS have one advantage, because someone asked, I think during the session, that a library, the availability of library. You know, in case of IR, the FTIR, there is a library available of gelatin. You can detect the gelatin based on the comparison of the IR library. Similarly, in case of GCMS, there is a library available of hundreds and thousands of compounds from NIST, and you can identify the compounds with the help of library very efficiently, right? It's no matter whether you have a high resolution. With the help of low resolution data, you can compare and identify the compounds with the help of libraries, and that is the basic advantage of GCMS. In LCMS, the people are trying to develop a library, including my own research group. We are trying to make a universal library of a small molecule. Some libraries are available, but in particular discipline, there is still lacking, right? So that is the important thing about the LCMS and GCMS. And please feel free to ask if you have any question in between, because I'm your live speaker. So don't need to wait till, you know. So, excuse me.
I'm just curious about the cost of LCMS.
Yes. You know, this is very important concern that everyone is curious about the cost of LCMS. Are you talking about the analysis cost or itself instrument cost? Instrument. It's this, yes, this is indeed a high-tech instrument and a very expensive instrument, no doubt about it. There's no doubt. It's a very expensive instrument and a high-tech instrument, but you know, we have to select our priority. If you really want to work something good, we have to purchase an instrument like LCMS or NMR. Believe me, without these high-tech instruments, you cannot produce a good quality research. That's what I believe. You have to have an instrument like an LCMS or MALDI. You know, we have to have in almost all institutions, because that is the future. If we want to compete with the world, we have to have all these facilities in all center institutions and keep it running. So it's a cost generally, अगर आप बात करें तो $200K starting from. This is a general cost of a normal mass spec instrument. It's not no matter whether it's a GCMS, starting from $200,000 and up to 800,000 US dollar. This is the general mass spec range instrument. The sample, as you know, someone, I think Dr. Rashid rightly said that the cost is always dependent that number of samples. So the mass spec cost is indeed comparatively high, but it depends that the number of samples. So the people are using mass spec, but you know that they are using mass spec when there is a need. If you can analyze the things on HPLC, then you should not move to mass spec, please. It's a word of caution. So preferably to analyze the things by HPLC if it is possible. If not, if really needed, then you should go to mass spec. Okay, so that's the important thing that you need to decide. So let's talk about the very quickly in halal domain. We can classify the things, different molecules in 3 domains. The deduction of, you can see the DNA. The people, you can see that the speakers are mostly talking about the DNA deduction. It could be a pork DNA or some other forbidden species deduction through DNA. So DNA is one of the important entity or molecule in halal research. Protein is the second important component, because we can see that the protein, especially derived from porcine, like gelatin and other stuff, that's one of the important molecule, you know. The third one is the small molecules, because we can classify the in halal research the three ways. There's small molecules, like it could be fatty acid in lard deduction, it could be a dye which actually derived from the insects. So you can classify the 3 types of molecules accordingly, and definitely in each case the techniques would be different. So as I have mentioned that mass spectrometry is quite in halal research. So since yesterday, I think you are watching different papers related to the application of mass spec. This is one of the example. You can see the lard deduction. But these are not from my research group, but some other one. So you can see that the deduction of lard with the help of time of flight mass spectrometry. Another paper, the gelatin deduction with the help of mass spectrometry. This is again kind of protein deduction, which you can see that is a combination, combined use of PCR and mass spectrometry board. Another example of detection, something forbidden substances with the help of mass spectrometry. LC-Q-TOF MS application in mass spectrometry. Maltitol is really important, especially if you are going to talk about the gelatin part. So gelatin is something which you can efficiently deduct with the help of MALDI. It's a quite high throughput. So the mass spec is there. So these are few examples of application of mass spectrometry in halal research. There are so many, I mean, but it just gives you an idea that if you have a mass spec instrument, so somehow you can cover the different aspects of halal research and testing in your lab. So as I have mentioned that initially I've started work on different food samples, then I've entered into the halal research. So you can see that this is an example of chickpea. Chickpea, Pakistan is one of the important importers of chickpea to the rest of the world, and we indeed have a very good and different variety of chickpea in Pakistan. But the problem is that because I have discussed this initially, I have no idea that what's the problem related to Pakistan agriculture crops. So when I visited Faisalabad, then they have highlighted that because of the drought condition, and which is nowadays getting higher and higher, they usually face a low crop yield, which is very obvious. And now they want to develop, the drought tolerant or drought resistant crop with the help of some gene mutation. But the problem is that they cannot analyze a compound which can protect the plant from drought resistance. And these are normally referred as osmoprotectant. They don't have a kind of analytical tool or the methods by which they can look. So then I just chip in, I said, okay, then we can analyze these samples. So one of my collaborators have sent me the samples who grown in a drought area. The another one is the normally the water availability area. Then we have compared their untargeted metabolomics profile. So this is a kind of slide where you can see a comparison of different metabolites, because we have identified so many metabolites, but you can see that we have listed around 20 metabolites which show a kind of upregulation or downregulation in two species which are grown in the drought or in the rain-fed condition. And you can see they are quite well differentiated. Some metabolites were appeared in high regulation, some were down-regulated, and those compounds actually are the compound which provides the osmoprotection of crop, and they can retain the water more and more. So this is the kind of a data which we have provided to our agriculture scientists, and they feel very happy to see the data because the changes what they have done genetically further proved by metabolome analysis. Okay, so this is the data which they need to know in order to get the confidence of their variety which they are introducing to the market. ये बहुत important data होता है जो कि ये link. So that was the link that we have made, with the agriculture scientists. The details you can see, it was just published, I think, last year in 2020 in scientific reports. Another sample that we have selected, it's about the crop. It's a rice, because we all know that the Pakistan is quite famous in aromatic rice varieties. But again, the same issue, they have a data of aroma, but they cannot link these metabolome set with their genome data set. So that's what we have tried to develop. We have conducted the extensive metabolomic analysis of different rice varieties and linked with their genome pattern, and somehow we were able to identify and differentiate basmati rice variety on a very characteristic metabolites of the non-basmati rice varieties. So I'm not going to talk about the details, as I have mentioned that you can look in the publication, but the purpose is to show that somehow you can, similarly mango, the mango is, we all, really like mango. So the important study which I would like to mention, that the artificial ripening of mango. Because metabolomic study I'm not discussing here, but you know that normally the supplier derive the mango with the help of calcium carbide, and we all know that the calcium carbide is really carcinogenic for your health, and that is the reason that sometimes we face a ban to export the mango, you know. So that's the issue. So we try to develop a method by which you can detect the contamination of calcium carbide on mango, whether it is derived by artificially or it's a naturally. So then we have conducted metabolomics analysis, a comprehensive metabolomics analysis, and what we have observed that that some metals like iron, cobalt, cadmium, and lead, which showed a clear differentiation and can be used to differentiate a mango ripe naturally or ripe by calcium carbide, and that is already published. But you know that what was the issue when we tried to validate our finding, because that was conducted under controlled atmosphere. So when we purchased mango from the local Thela and all these things, because of the environmental pollution, this study totally failed because they already had a contamination of so many metals. The concentrations were too high. So at that case, the study is still not valid to check the commercially available, you know, the Thela stuff and all these things, whether it is derived by calcium carbide or not. But at least it's a study by which we can see that how these metals are getting raised and used for the differentiation. Honey adulteration is one of the important issue that especially in Pakistan, the people likes honey, and in the Western world as well. The one of the adulterant, the most common adulterant which people are using to adulterate the honey is brown rice syrup. किसी ने देखा है brown rice syrup? Have you ever seen that or not? Believe me, you would not be able to differentiate whether it is honey or rice syrup. It exactly like honey. Its taste, its texture, its smells, everything is like honey, and you may take it as a honey, but this is a brown rice syrup, which means that it's nothing but sugar content, a complete, no physiological property of honey. So when I saw that this brown rice syrup, I said, oh, this is honey. My student said, no, sir, this is brown rice syrup. So then I realized that it could be adulterated in honey samples. So then we have collected around 30 or 40 honey sample all over in Pakistan and tried to analyze. And believe me, the 2 samples were identified as a contaminant, heavily contaminated by the brown rice syrup. And this method was developed with the help of NMR spectroscopy, because you can see that in this is a comparison of ABC. You can see it's a simple original honey, commercial available honey, and you can see that there is an additional peak. This part, this is an additional peak which is related to brown rice syrup. And you know how the brown rice syrup is formed? They usually subjected to the various enzymatic hydrolysis, the partial hydrolysis of rice. So once it's partially hydrolyzed, they just cleave into oligosaccharides, including disaccharides. So this disaccharide is actually a characteristic marker for the detection of brown rice syrup in honey, because in honey we don't have such type of thing. So this is the method, and the people, especially in Australia, the people are using the same method, because I am in touch with them and they asked me initially, and now they are using the same method, and they are detecting a contamination of brown rice syrup in honey with the help of same method. So this is the thing, and it's already been published. Now come to the halal part very quickly. The important thing that we all can identify, ये गधे का गोश्त है और कुत्ते का गोश्त है और किसका गोश्त है, with the help of DNA. We can easily identify whether what meat it is, but we cannot so far. I mean, there is no method available by which you can differentiate zabiha or non-zabiha or dead chicken. आजकल इतना ज़्यादा डेड चिकन के इश्यूज आए हुए हैं कि personally I'm also scared whether I'm eating a dead chicken or the zabiha one or not zabiha. Anyway, so the first part of any commercial testing, it's a basic research. You need to conduct a kind of basic research or identify molecules by which you can develop a test, okay, through by which you can differentiate whether it is zabiha or non-zabiha or dead meat. So for conducting this research, a very interesting story I just shared with someone yesterday, that when we're trying to work on this Zabiha part, so the first important section was to get the ethical clearance. Ethical clearance for this type of work is nothing but the fatwa, because I don't want to kill the birds for my own research, because it's important issue. So then I've sent the whole story that I want to cut few chickens in a non-Islamic way, so kindly allow me. I sent all this detail to the Islamic scholars, and they were very upset. And they said, what type of fatwa he's asking for? So just sit and discuss. And after 1 month, they just provided me a fatwa that, okay, for the sake of research, you are allowed. So that was the interesting thing. But anyway, another issue that, another hurdle that we faced, that no butcher was agreed to cut the chicken in a non-Islamic way. And they are scaring that, are you going to do some kind of jadoo or kya, why you want to have such type of slaughter method? So my student, I asked my students, can you do all this slaughtering by yourself? So he is the one who actually cut the chickens in a non-zabiha way, because zabiha we just got that from the butcher house, and the non-zabiha and the dead chicken, because the dead chicken, they usually, once they arrive to the poultry farm, from poultry farm to the butcher house, most of them unfortunately are dead. And they used to have that chicken as well. It's very unfortunate. So we collected some dead chicken. That work is under progress, because the dead chicken sample are more complex to handle. So anyway, so these are the 3 groups that we have included in our study. And if you just look, this is the LCESI Q-TOF MS spectra. It looks quite complicated. If you just look these spectra of 3 groups, the DAT, the non-Zabiha and Zabiha, they look quite similar, except the difference of their intensity spectrum, okay? Because that we have identified through detailed research. And now you can see that the volcano plot, if you just see the all metabolites that we have identified on that base, you can see these 3 groups can be differentiated very efficiently. All these 3 groups, you can see that we can differentiate based on its high resolution ESI, MSMS analysis. That is a good sign. And once we saw this result, so we felt happy that we are moving in the right direction, and the compounds are there by which you can differentiate. So these are the 5 most important compounds by which you can differentiate the Zabiha meat with the non-Zabiha meat. This is actually the findings, and you can see that we have identified the compounds in a very high accuracy. As you can see that the threshold values are quite low. So we have identified the compound with very high resolution. And believe me, the every molecule has its own story. Very shortly, what I would like to tell you, for example, in case of linolenic acids, one of the important fatty acids. And this fatty acids, in case of Zabiha, this is a very important nutritional fatty acid. And this, in case of Zabiha, this fatty acid is higher in concentration. We, being a Muslim, we believe that the Zabiha meat quality is good, but good not in terms of hygiene, in terms of nutritional point of view as well. Because in non-Zabiha, the blood retain more in the tissues, and due to that buffering, due to that blood, they extract the nutritional compound in it. So the compounds which has a high nutritional value found more in Zabiha in comparison with the non-Zabiha. So similarly, all metabolites have very interesting story, and some metabolites are also associated with the stress trigger associated pathways, which means that actually is further under research, which means that the Zabiha, the animal who is going to slaughtered by Zabiha, feeling a less pain. That is a big debate nowadays, that whether they're feeling a less pain or the more pain. So the science somehow will prove, because we have identified one of the molecules which links with that pathway. So all these 5 molecules, you can go to the journal paper and look and read in a detail, and you would be happy to that what type of I'm not going to talk about the identification levels for the students. So these are the set of metabolites, because the 5 compounds that I have shown were very important to differentiate the zabiha and non-zabiha meat. But apart from that, these are the different metabolites, a set of metabolites that can also be used to differentiate zabiha and non-zabiha meat with very high accuracy. So this is a kind of chromatogram that we have observed in individual samples, the upregulation and downregulation of similar metabolites in both zabiha and non-zabiha samples. And this is, you can see downfold changes and up and down regulation of these metabolites in terms of their concentration in zabiha and non-zabiha. You know, the validation of the study is very much important, because once we have conducted the complete analysis, then we have subjected few samples randomly. You know, we just conducted a kind of blind analysis that we just mix this zabiha and non-zabiha sample and ask my student to analyze all these samples. So once they analyze and classify based on our developed data, and once we decode, then you can see that the overall accuracy of the method was too good. It was a kind of method by which you can differentiate the zabiha meat and non-zabiha meat. The question is come to, you know, the last, I think yesterday, Dr. Isra was rightly mentioned that we need to have tools which can be used on halal site detection, you know, because it's not that easy to analyze these samples on a routine basis using such type of methods. But as I have mentioned, that every study requires some basic study as kind of, you know, the basic study. So this is the kind of study which can lead to the research where we can develop a tool or sensor by which we can differentiate the zabiha and non-zabiha food samples. This is, I think, not in shortage of time, I would not talk about. Yes. A kind of, yes. Actually, this is the molecule which show a drastic difference. You know, there is an always important whether you want to see how many folds of changes. That is the thing. The molecule which we have mentioned is the one which actually show the maximum fold changes. Otherwise, the biomarker, not the biomarker, but the marker that we have identified were 6 in number. So those 6 panel biomarker can be used to differentiate. But just as a, you know, in a graphical abstract, we have mentioned that this is the one which can be used for, you know, for clear differentiation. It's indeed the higher one. Yes. No. It's a phosphocutidyl kind of molecule. It's not a lysine. Actually, the reason exactly don't know. I mean, like, yeah, because, you know, they link to the various metabolic pathways. So this, once we identified, then we're trying to link a reason, just like I've mentioned that in case of fatty acid, it could be reduced because of the more blood retained in the muscles. It's just a hypothesis, but we have identified the compounds. The exact reasons need to be validated. You know, it definitely takes time, but at least now we have identified molecules which can be further proceed, which can be further studied. Okay. So, you know, you have already heard about the analysis of gelatin. I'm just going to give you a picture of Maldi-Toff a snapshot, because the Maldi-Toff is a kind of instrument which gives you a rapid analysis, maybe less than a minute. So the technique, which is very sensitive and can be used for the differentiation of gelatin of different origin, because the last speaker was discussing about the identification of gelatin with the help of LCESIMS. LCSIMS indeed the good and sensitive as well, but it's not a rapid because it takes time to prepare the sample and everything. Maldi is a kind of instrument, as obviously is a sister instrument of mass spec, but it's a kind of technique by which gives you a rapid identification. So this work is going on, that we are trying to develop. We have identified some pattern, but it's under research. So most probably in the next event I would be able to. So this is my research group working with me, standing with Professor Iqbal is also serving as a director ICCBS, apart from coordinator general of comstech. And these are the students who actually worked on such type of issues that I have mentioned. The work is actually funded by a different funding agency, which is very much important. If you would see this R&D, it's currently known as Ignite. अगर आप में से कोई कंप्यूटर का कोई बंदा है तो they must know that initially. So Ignite is a kind of IT based funding, and I am the only chemist who got the funding from from ICT R&D. You know, when I was presenting that work in front of his ICT director, he was laughing before my presentation. He said that I'm surprised to see a chemist and defending his project in front of IT people. So they were all IT people, and I was the chemist, and I defend that and got that project. So the reason to mention this thing that you can also get the project. So it depends on how you design the project. Okay, you can introduce some IT part that can defend very easily. Higher education, the OPCW is one of the organization, international organization, Nobel Prize winner organization who used to fund the research and training as well. So those who are working, I mean, especially students, they can apply to OPCW. The work, especially to Zabiha and non-Zabiha, was actually funded by Islamic Development Bank. As you have seen that Professor Iqbal mentioned that we have developed a lab in the Kuwait Scientific Institute. So I was the one who actually had this whole project and got the funding from Islamic Development Bank. And now the KSR has a fully functional halal testing laboratory assisted by us by the ICCBS. Very shortly, ICCBS, because I belongs to ICCBS, so for the students, important to know that what facility we have. We have a number of instruments, including 800 megahertz NMR, the X-ray facilities for structural elucidation, dozens of mass spec available for such type of analysis. Similarly, we have a cell culture facilities, imaging facility for tissue analysis, the patch clamp for neuropharmacological work, molecular and genome center from where Dr. Ishtiaq belongs. So we have high sequence next generation instrument for quick genome sequencing. The computational facility, we have a computational biologist, computational chemist are doing wonderful work. Huge library updated with various research journals and abstracts. We also have a tissue culture facility, and we used to provide these ornamental plants to the rest of the hotels available in Karachi, because we used to grow. This is a commercial wing. We provide a commercial testing and some pilot plant for the herbal companies for their industrial scale extraction. So, addition to that, we provide accommodation to our researcher, to our speakers, to our guests, and we also have a nursery as well for children. Okay, so for those women who have kids, they can bring their kids, and they used to. I mean, we have nursery in all buildings, so we actually encourage the women to come. We have a points facility to go, to some sports facility. Our brilliant students, foreigners are used to visit us very frequently. Some of them you can see at the back. So you just grab them and ask them to just join us for so they are there for training and a different program. And this is our group photo. So with this, again, thank you very much to all of you and your kind attention.
Thank you, Professor Musharraf, for your detailed talk. As we are running short of time, so we can have quickly one or two questions, please.
What I would recommend, yes.
Most of the time, our product, we are much focused on the protein profiling.
So do you have any experience while using the 2D gel electrophoresis? They can give us a whole profile of the proteome, and after that going towards the multi-top. The second,
yes, please.
so the second question is, it may need like a bit of compassion and empathy. While studying your the slide you showed about the Zabiha and non-Zabiha. So I'm just curious to know about the sound and concrete proof for the technical justification, scientific evidence like. So Edward, do you feel as it would like to slaughter these chickens from a non-Muslim following the same practices and to make a comparison between these groups? And one another question that how many groups were used in this study, Zabiha and non-Zabiha?
Right. We are very much in as far as the proteomics analysis, including 2D dye-jet, 2D gel electrophoresis, but not for the food samples. Because my research group is working on different biological samples, particularly plasma and serum. So we used to have such type of separation technique followed by mass spectrometry. But as far as the halal concerned, we have not analyzed the things with the help of 2D dyes or such type of technique, because the paper is already in the market. The people have already compared a similar group with the help of protein, and they identified some differential protein as well. So as far as the capacity concerned, technical expertise concerned, yes, we do have. But so far, I've not applied this protein, such type of approach towards the halal's part. Second question was related to the control group. The control group, because the important thing, if you want to compare the two things, that everything should be similar. Then you can compare the metabolites pattern. So we try to make sure that the chicken who are taking from the same same farm, getting the same feed, the gender was same. Their age was same, so that what we have tried to make the control similarity as much as possible, except the difference of slaughtering. So that we have tried to, because otherwise you cannot compare the group together. The third part, as I have mentioned, that these are the preliminary study, and it's a concrete study as far as the metabolites identification confirmed. We have identified the metabolites at the stage level of 2. So the important thing is to just follow up further and to validate on a large number of samples, probably 200 or 300. And we are looking forward to do the same thing, but we require a kind of huge funding in order to conduct such type of things. Actually, we are running indeed short of time, so I'm available. Feel free to ask me during the lunch if you have any question. Okay, thank you very much.
Our next speaker is Dr. Muhammad Ali Al-Sheikh Wais from Turkey. He is an associate professor of clinical pharmacy in drug information domain. He got his PhD in clinical pharmacy in 2014 from University Science, Malaysia. As halal pharmaceuticals and halal cosmetic scientist, his work and research focus on formulation adjustment, halal alternatives for doubtful ingredients, halal critical chemical analysis, halal enzymes and culture media solutions, and halal drug information. Dr. Muhammad Ali joined SMIC in 2018, and he is also the convener of the Porcine Detection Experts Working Group. So over to you, Dr. Muhammad Ali. Dr. Ali, are you there, please?
Assalamu alaikum. The voice is okay?
Yeah, it is okay, fine.
Can I share my PowerPoint? Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning to all participants in Pakistan and all friends in online session. Inshallah, today we will speak about the animal-based ingredient in cosmetics and about their analysis technique. I will focus more on the cosmetic part, the ingredient part, and the halal critical points, inshallah, in this presentation. So generally, we will speak first about cosmetic ingredients in general. What is the trending in ingredients sometimes happen? What is the animal-based ingredients? The critical point for the halal critical point for the animal-based one, the biotechnology ingredients and the halal critical points. And at the end, we will speak about the type of test needed and the official tests in some country regulations. Just to give you an idea about the cosmetics, it's different than pharmaceutical and food. Cosmetics industry depend on trending. There are thousands of chemicals. Literally, there is 1,000 of chemicals, and there is 100 of chemicals added every year, because this industry depend not in the proof evidence-based proof. It's depend on the claims. So you can add material and you can do claims, and the market allow you to market without evidence on this product. So in general, we have many chemicals added every year to the ingredients. There is international system, nomenclature system for the cosmetic ingredient, the INCI one. It's the system used to give short name for each chemical used in cosmetics. Since we have thousands of chemicals, we need short name, not like the systemic chemical name. You know that the IUPAC chemical name system is very long sometimes. So they use the INCI name in cosmetics. The INCI name is produced by the Personal Care Products Council, which is used to be Cosmetic Toiletry and Fragrance Association before this time. The Cosmetic and Toiletry bench reference disk is the most wide reference used for to get information about this cosmetic ingredients. It was published before this time in printed book. Now it's available online. So you can go there and do search, and you will find thousands of chemicals. You will find some information about the sources in some cases, but unfortunately, you cannot find every time information about the animal origin or about the biotechnology origin sometimes. So this is where we need halal studies. There is thousands of chemicals. They put the name of the chemical, they put the composition of the chemical, but they don't put the origin of the chemical sometimes. We need to do some studies to investigate the market and know which one come from halal source, which come from non-halal source. Another important website to find information, free website, it's the Special Chem website. It's also contain the majority of the INCI products inside it, with some formula and with some perfume database, which help the cosmetic people and the halal auditor to know more about the ingredients of the cosmetics. So in general, the raw materials of cosmetics can come from chemical synthesis or from plants. Until now, the chemical synthesis is the majority, but there is a trend now to reduce the chemical product in cosmetics, because the market now going to market the cosmetics as green, as environment friendly. So they start to reduce the chemical synthesis product, And the problem with the plant product, the botanical products, that it's differ from one country to another country. The plant and the extract concentration differ from one soil to another soil. So you can get the chemical, but it's not the same standardized extract every time. And this is makes some problem in the production of cosmetics. As you know, the plant botanical source is halal, and the chemical source, if the process don't contain any najis material or non-halal material, usually it's halal. So these two sources, usually we don't have big problem in halal with these two sources. The problematic raw materials come from the animals and from the biotechnology source. People used cosmetic from the before history time. We don't know when they start to use cosmetic, but as we know the history, women used cosmetic from long times. We have records for using cosmetics from the old Egypt and Iraq around 7,000 or 8,000 years ago. One of the most important animal ingredient, which is still used until now, the musk, which is produced from the musk deer, the ambergris, in Arabic we call it amber. It's produced from the sperm of the whale. We have the gift cat and the castoreum from the beaver. All of these type of animal byproducts, these types used in cosmetics until now, especially in perfume and flavor industry. Actually, when we start CIMIC 24 about food additives, we find many flavors and many perfumes come from animal source, but it's limited to this four and their biotechnology products. They take the main perfume from the animal and they make some modification by biotechnology and they use it after that. Some of the famous animal-based products in cosmetic industry is the wool fat, lanolin, collagen, elastin, and beeswax. All of them used in skincare creams. We don't have problematic issue with the beeswax, and it's halal, but also for the lanolin, the problem come from the collagen and from gelatin materials. There is some anti-wrinkles creams. It's contain various animal proteins like bovine serum albumin, oval albumin, big placenta albumin. All of them, they used to tighten the skin, which give it younger look for the skin. We produce some cosmetics from fish, like the nail polish and the glossy of the mother of pearl. All of them come from fish side. Also, we don't have problem with the sea products in halal status. In CIMIC 4, it's give the main halal status and the halal important steps to produce halal cosmetics. So I'd like to highlight two things from CIMIC 4 regarding halal cosmetics and the animal products. The insect status of halal status of insect in CIMIC OI4, CIMIC IC4. Actually, in CIMIC 1, insect is not allowed 100%. It's considered as non-halal to consume insect. But in CIMIC 4, it can be used for external use if it's come from a clean environment and it's eat clean material. This is the two conditions that's mentioned in CIMIC 4. So, for example, if there is insect and it's what's called grow in environment which is clean, not sewage, not dirty places, and it's good material, so we can use it in industry. For example, carmine is not allowed in food as coloring agent, but it's allowed in OIC CMIC 4 in cosmetic preparation. Also, I'd like to highlight about ethanol. If you remember yesterday, I speak about ethanol. In cosmetics, it's allowed for external use. In food, it's not allowed. So this is two important topics in cosmetic differ than food. And both of insect, some people say that it's not allowed in food, but it's allowed in cosmetics. CMIC TC2, the halal cosmetic issues, is working now on the animal-based ingredient doubtful lists. It's new standard project, still in the beginning, but we are working in this animal-based ingredient doubtful list to list the major animal ingredients in the cosmetic industry. If you allow me, I will share with you part of this list. So in general, this list contains around 200 chemicals. We make some research about the origin of the animal-based ingredients, and we found around 200 chemicals. We try to find the different sources, bovine sources, non-bovine sources. When we write non-ruminant, that's mean porcine. We found from poultry, also from botanicals and synthetic sources. We depend on the FDA database. Since the FDA sometimes mention the source, it's the only organization which we found some database we can extract the information about the origin of the chemical from there. So you can notice here that there is a lot of lanolin product. We don't have problem with lanolin, but it's part of the animal products. There is a lot of fatty acids, many different fatty acids, a lot of extract. We have brain extract, thyroid extract, aorta extract. This extract is used in different kind of cosmetics material. Collagen, you can find many collagen and collagen amino acids in this list. The amino acids, we have problem also with the amino acids, since there is different amino acids used in different cosmetics, and we have around 6 or 7 amino acids problematic, since it's used a lot in cosmetic, and the majority of them come from animal source. We have the tallow product. There is around 15 products come from tallow, and as you know, tallow is the collection of extra meat and extra fat after slaughtering. So they take this extra tallow and they grind it, and it's sent to another factory to make extract and to melt it to be used in other industries. All gelatin types, we calculate around 15 different types of gelatin. And as you know, gelatin has a special case. We will speak about it now. And glycerin, it's also very common. We find many milk products. It's halal, but some of them is treated with special enzyme, which make them different products. And it's need also for check the halal source of the enzyme that they use to change this enzyme, to change this milk. So as you see here, also we have lard, and there is different byproduct of the lard used in the cosmetic industry. Elastin, different kind of milk protein treated with different enzyme. So the problem not with the milk, but with the enzyme that they treated of this milk. Extract, you can see here, mammary extract, marrow extract, liver extract, all of them used in in this industry. So in general, this list until now, until study in CIMIC TC2, inshallah, it will produce as CIMIC standard. Maybe not in near future. It will take around 1 year or almost 1 year and a half. But hopefully, inshallah, it will be full detailed list, which is not available in the market until now. Most of the people who is going for halal auditing until now, they don't have a list help them to search for the origin of the chemicals in the cosmetics. So inshallah, it will be in our project in CIMIC. Come back to the slides. Okay. So it's important to check for the regulations sometimes about the cosmetics and cosmeceuticals, and to know the difference between cosmetics, food, and medicine. In CIMIC 2, we study only cosmetics. Now we have TC16, which is study pharmaceuticals. And why I'm speaking about this one, because the halal status will be different according to the usage of the ingredient. For example, cosmeceuticals are cosmetic products with bioactive ingredients. What's the difference between cosmeceuticals and pharmaceutical? Cosmeceutical includes some active ingredients, but it's only claims. There is no evidence for this one. They have some used, but it's only claims. While pharmaceutical, it's indication and it's used for this purpose that they put it. So what's the difference if we mention that this is cosmetics or it's medicine? This is food or it's medicine? It's important in our halal status, because we allow sometimes some non-halal material or some najis material because of the darura, the necessity to use this chemical. When there is no darura, we cannot use it as cosmetic, for example. I'll give you an example for this one. The hyaluronic acid. Hyaluronic acid is produced from different types. We have many origin of hyaluronic acid. It can come from the umbilical cord of human being. It can come from the rooster comb, and it can come from the biotechnology source. The majority of the hyaluronic acid in the market is from the biotechnology, and there is around 15 to 20% come from the rooster comb. So there is some medical uses for the hyaluronic acid. It can be used for injection, for the knee, especially for the people who has problem with the knee, and also it can be used to treat some eye disease as injection, also ophthalmic injection. We have famous brand of medicine. It's called Hyalon. It's also used for medicine. Also, it's used as cosmetic. But the majority of the market now is in the cosmetic part. So when we go and study hyaluronic acids, it's come from these two sources, from animal source, from the rooster comb, and we need to check the halal status of this rooster comb, or from the bacteria. And both of them, they have some critical halal point, which it's not halal 100%. So if you say I'm going to use it as cosmetic, it's not like you are say I'm going to use it in medicine. In general, after we speak about the different animal ingredients used in cosmetics, there is some critical points to produce this animal based. When you go for halal auditing, the most important one, we follow the OIC CIMIC one, the food things. The halal starter houses is the most important one. When we study the list of the critical ingredients, we find that not the porcine is the most used animal. The most used animal is the beef industry. So for example, we have 250 chemicals, only 30 of them come from porcine, and around 150 come from beef industry. So the critical one is the beef industry. We need to check the slaughter condition of this beef to make sure that it's halal. There is another concept also we should know about Simic opinion about istihala. You know, there is in some fatwa, they mention that if you take one chemical and it's changed to another chemical, 100%, you can use it according to the istihala. And some people misuse this concept to allow gelatin from pork, to allow gelatin from porcine sources, and to allow gelatin from beef, which is not slaughtered according to Islamic rules. So in Simic standard, it's very clear. The animal must be halal, and it must be slaughtered according to the Islamic rule, which exclude this istihala rule in this misuse that some people use it. So we cannot use gelatin from pork. We cannot use gelatin from halal animal, which is not slaughtered according to the Islamic way. And inshallah, Simic 22 about halal gelatin will publish soon, inshallah, and it will have full details about the production of the gelatin. But still, gelatin until now one of the most problematic animal ingredient in our cosmetics and our food and in our pharmaceuticals. Gelatin, as you know, it can produce by two methods: from skin of the cows and also from the bone. In Turkey, we have 4 gelatin factories which produce it from skin of the cows, and from the bone it's produced mainly in Pakistan. So until now in Islamic countries we have limited number of gelatin factories. I think around 14 factory according to our latest study about gelatin factories in Islamic countries. The majority of them in Pakistan and in Turkey. There is 1 factory in each of Iran, Egypt, Indonesia, and Saudi Arabia. This is the each country have only one factory. So alhamdulillah, we have some country which produce gelatin, but it's not enough for the consumption of the Islamic countries. Okay, there is one question every time when we speak about animal ingredients. People ask us this question: what is the percentage of the animal ingredients comparing to the other ingredient source? We don't have study. Unfortunately, we don't have studies. I have some old studies about this one, but we need to make a special study about the origin of the ingredients in the current days. And inshallah, this is in the plan of Simic, inshallah, in the near future. So for example, glycerin, as you know, glycerin, one of the most used ingredients in cosmetics. So when we check for the glycerin sources, we find that it's around 30% animal base, 70% is petroleum and botanical sources. So there is around 30% doubtful material around the world. You can see here in this table. Also, I find some information about the world production of fat oils. So you can see here from 100%, there is around 20% or 18%